Disaster Trio.
Hi.
Hola,
Cześć.
welcome to another episode from the Disaster Trio. I am your host, Francisco. First, a full disclaimer. At the moment, I work at the University of São Paulo, Brazil, as a postdoctoral researcher in the development of biopharmaceuticals. The following episode is mostly based on the book The Cutter Incident by the medical doctor Paul Offit and my own experience with biotechnological process development. We're recording this episode in July 2020, during the coronavirus pandemic, and many discussions are now happening online about the pharmaceutical industry and the safety of vaccines. Unfortunately, many of those discussions are based on a poor understanding of an industrial production of biological pharmaceuticals, as well as medical science in general. Therefore, we believe that a proper in-depth discussion of the Cutter incident can help you to better understand how vaccine development works and hopefully shed some misconceptions along the way. In 1916, the United States was facing the first major outbreak of poliomyelitis. It started in New York, and by the end of the year, more than 9,000 people had been paralyzed and 2,400 people had died. Much in the same way as in the current coronavirus pandemic, many people did not understand what caused the disease. They blamed animals, pasteurized milk, wireless electricity, radio waves, doctors' beards, and poisonous gases from Europe, whatever those are. And they also blamed parents for tickling their children too much. At the same time, the poliovirus had already been studied and its mode of transmission was well known by virologists and medical doctors. People also tried many different cures for the virus. They ingested catnip, blackberry brandy, and the blood of frogs, snakes, and horses. They also hung charms around their necks made of pepper, garlic, camphor, and onions. And of course, needless to say, none of these strategies helped much preventing the spread of the poliomyelitis virus. Unfortunately, most people who contract polio never develop any symptoms, which helps to spread the disease to their friends and loved ones. Unfortunately, much in the same way as the coronavirus is doing nowadays. It is therefore no surprise that the efforts to quarantine only the sick people failed to prevent new polio outbreaks in the US by the beginning of the century. So by 1934, researchers already knew that people who were infected once, they were immune from new infections of poliomyelitis, so they started working on the first polio vaccines. Dr. Maurice Brody tested the first inactivated vaccine. Unfortunately, his trials resulted in several polio cases, which scared researchers from working on this topic for almost 20 years afterwards. It took the creation of the National Foundation for Infantile Paralysis with the support of perhaps the most famous polio victim worldwide, which was President Franklin Delano Roosevelt of the United States, in order to gather an unprecedented amount of money to research polio and to study the development of a possible vaccine. Several researchers, who unfortunately I will have to omit here for the sake of time, worked really hard in many universities and laboratories for a really long time in order to find how polio infects the central nervous system and how does it cause paralysis. They also studied how the virus could be safely grown in a laboratory and in large quantities, and how it could be then inactivated in order to create a safe vaccine. So probably the name most associated with the development of the first polio vaccine was that of Jonas Salk. Although Salk did not single-handedly create the polio vaccine in his lab, it is true that he was central in organizing the research efforts and that he contributed significantly for its development. The Salk vaccine was produced by cultivating monkey kidney cells in bottles in a laboratory and then infecting those cells with live virus. The virus then multiplied and ravaged those cells, leaving the liquid in which those cells are suspended completely filled with active, infective viral particles. This liquid was then filtered and the viruses were inactivated using a small concentration of formaldehyde during several days. So in 1954, the first clinical trials of the Salk vaccine began. An unprecedented 420,000 children were vaccinated, and the results showed that the vaccine successfully reduced threefold the number of children paralyzed among the ones that received the vaccine. This result was immediately reported by the media and became a sensation. Jonas Salk was the star scientist at the center of the success. Encouraged by the initial results, the American legislators pressed the industry to rush the production of large amounts of the Salk vaccine for the National Immunization Program. By 1955, less than 1 year after the first initial clinical trials, 5 million doses had already been produced by 5 companies: Cutter Laboratories, Eli Lilly, Wyeth, Parke Davis, and Pittman Moore. After a few weeks from the start of the vaccination program, the first reports came in about children developing paralysis after the vaccination. However, since polio was still endemic in the United States at the time, and the vaccine was known not to be 100% effective, it was of course possible that those cases they were the result of the natural circulating virus and not from the vaccine itself. However, only 48 hours after the first cases were reported, statistical analysis showed that the cases of paralysis were indeed associated with some of the lots produced by Cutter Laboratories. Also, the fact that, in some of the cases, the arm that received the injection containing the vaccine was paralyzed was evidence enough that the poliovirus present in the vaccines from a few of the lots produced by Cutter Laboratories was not always completely inactivated. So, the United States government suggested Cutter to recall its vaccine lots. And Cutter complied, and within only 38 minutes, they had sent out a telegram asking all of the health departments and drugstores to stop using their vaccines. By the end of the incident, at least 220,000 people were infected by polio from the Cutter vaccine. 70,000 people developed muscle weakness, 164 were severely paralyzed, and 10 people were killed. So how could a product that was designed to prevent polio be causing it? The answer can be summarized by a series of 7 events, which all needed to have happened exactly as they did in order for some of the Cutter vaccines to be contaminated with live viruses. First, the polio vaccine formulation needed to contain all 3 main types of virus in order to be effective, which were creatively named types 1, 2, and 3. Jonas Salk used the so-called Mahoney strain to represent type 1 polio in the vaccine. This strain was particularly virulent and very hard to kill, making the inactivation process very, very delicate. Salk knew that, but he chose this strain anyway because it was also particularly good at generating a strong and lasting immune response in the patients. As long as the inactivation was done properly, this would not have caused any problems. Indeed, the current inactivated polio vaccines are now safely produced using the Mahoney strain with the help of a carefully designed inactivation process. The strains that were used to represent the other types of poliovirus in the vaccine did not cause any of the infections. Second, in order for the inactivation process to work, the virus culture had to be first carefully filtered. The Qatar laboratories used glass filters for this purpose, which turned out not to be very reliable. And if the virus culture is not crystal clear before being mixed with the inactivating agent, live virus particles can hide inside the cell debris and survive the inactivation process. Nowadays, very stringent requirements are in place to only allow production batches that meet precise clarity requirements to be further used in the production process. Third, the federal government only required 2 safety tests. Test number 1 was to inject the vaccines into living monkeys, and test number 2 was to mix those vaccines with monkey kidney cells, which were cultivated in glass bottles in a laboratory. If the monkeys were not paralyzed in 30 days, and if the cells growing in the bottles didn't die in 14 days, then the vaccine was considered to be free of infectious polioviruses. Unfortunately, this turned out to be insufficient to guarantee the safety of the vaccine for every single person that could possibly be vaccinated. For instance, a few people are immunocompromised, and very few active virus particles are enough to cause the disease in them. Afterward, the authorities have changed the official tests to use much larger amounts of the vaccine injected directly into the spinal cord of immunocompromised monkeys, and this everything makes the safety testing several hundred times more sensitive to potentially active infectious viral particles. Nowadays, much more reliable and sophisticated tests are available for the companies to be sure that they are providing a safe and effective product. Fourth, Qatar laboratories waited too long between the virus production and the filtration of the liquid. This caused particles to precipitate inside the bottles, which made the filtration even harder. Nowadays, there are very specific requirements about how long a company is allowed to wait between production steps in order to assure that their processes are very reliable. Fifth, Qatar laboratories did not follow the recommendations of John Salk to make a very careful mathematical model of the inactivation process. And this point is a bit tricky to explain, but it is the main reason why the tragedy happened, so it is worth taking some time to understand it. Basically, the inactivation process is very hard to predict, so Jonas Salk recommended that the companies take samples every few hours during the inactivation process and test the live virus concentration in them in order to know how the inactivation process is going. As I said before, very minor differences in the process used by different companies, such as the filter that they used or the waiting time between the filtration and the other steps, are enough to dramatically change the time necessary for the inactivation process. Knowing that, Salk suggested that the companies follow the concentration of live virus very carefully during the inactivation process in order to know exactly how long it is taking for the concentration of living virus to drop below 1 active infecting virus particle per dose on average. Afterward, the companies should multiply this time that it took to reach only 1 active infectious virus particle per dose by 3 in order to be really sure that there was no virus left alive in the vaccine. The problem was that in many cases, multiplying this time by 3 was not enough to completely kill the virus, especially if the virus could hide inside the monkey kidney cell debris, which were used in the production process, as it was happening, unfortunately, in the Qatar laboratories production. At this point, You might be asking yourself, why didn't Jonas Salk just increase the safety margin to much more than threefold and use a much higher concentration of inactivating agent to be really sure that the product was safe? But the problem is that if the inactivation process was too harsh, the vaccine didn't cause immunity. So the inactivation process had to be designed very carefully in order to kill the viral particles, but not destroy them so much that the vaccine would become useless. The solution proposed by Salk was a very precise engineering of the inactivation process, which Qatar Labs didn't do. However, the other companies that were making the vaccine at the time, they followed the recommendations from Salk and measured very carefully the reduction in infectious viral particles during the inactivation process. Because Qatar didn't have a good mathematical model of the process, it was having inconsistent inactivation results between the different production batches. Indeed, out of the first 27 production lots, 9 had to be discarded because they failed the monkey safety tests, and Qatar knew that. However, at the time, the companies were not required to inform the government about how often their process failed to completely inactivate the virus. So, from the perspective of the US government, the production at Qatar Labs was doing just fine, which is the sixth reason this tragedy happened. This is called selection bias, which is what happens when people do not report bad results, thus creating a wrong picture of how well they are doing. For instance, if you flip a coin many times, but you just tell your friend about the times it landed tails, your friend would be forgiven for believing that your coin has tails on both sides, right? This is more or less what the US government did when they did not order companies to disclose 100% of the results from their production batches. In fact, all companies that were producing the polio vaccine at the time were having a certain percentage of failed batches, and the US government was completely oblivious to this fact. For instance, Parker Davis made 234 lots, out of which 21% were not completely free of living viruses. Eli Lilly made 85 lots, from which 8% contained live viruses. Wyatt made 267 lots, 5% containing active infectious viral particles, and Pittman Moore made 126 lots, 2% of which contained living viruses. However, because these companies were using better safety tests than Qatar, they never sold the vaccines that contained living infectious viruses. Had the government officials known that the companies were having inconsistent virus inactivation results, they would likely have interrupted production in order to figure out what was going on and require more careful testing protocols and a deeper scientific studies to understand why the inactivation process was so often unsuccessful. Indeed, nowadays pharmaceutical companies are required by law in most countries around the world to disclose 100% of their production statistics, even about the failed batches that were thrown away anyway. This requirement allows government officials to be better able to judge if the pharmaceutical companies have their production under control. And seventh, the government did not require the companies to be able to produce several successful batches in a row. This is a standard requirement nowadays, but at the time it didn't seem so important for the authorities. Basically, if a company is able to successfully produce several good batches in a row, it means that they really understand their process and that they have it under control. This is important because the government doesn't have enough resources to investigate every single detail about every single process in every single company in order to be absolutely sure that everything is running perfectly well. The company employees are actually the people that are in the best condition to know what are the possible sources of problems in their processes and how to fix them. And the way that the companies can then prove to the government authorities that they indeed have everything under control is by being able to produce several batches in a row without a single glitch. Imagine, for instance, that you are making bread at home, but sometimes it grows, but sometimes it doesn't. Would you be absolutely confident that you know everything that is going on in your kitchen? Probably not. However, in the case of a failed bread, you can simply throw it away. But viruses, on the other hand, are very hard to detect, so you should only feel confident that you have everything under control if the production process never fails. As you see, the cutter incident only happened because a series of bad decisions were taken. It was necessary that everything went wrong exactly as it did, exactly at the same time for this disaster to happen. If even one of those hadn't gone wrong, the tragedy would have been averted. For instance, if the safety tests were more sensitive, the production problems would have been immediately detected and the lots would never have been sold. If the filtering process was more reliable, the inactivation process would also worked more consistently and cutter would not have lost any lots. Even with the bad filters, if cutter laboratories hadn't kept the virus cultures waiting so long before filtering them, there would not have been enough time for the precipitation to occur and the filtering process would probably have worked properly. And if the US government had required companies to share all of their production data, the production batches would not have received marketing authorization in the first place. And finally, if Jonas Salk had chosen another strain of poliovirus, different from the Mahoney strain, to represent type 1 polio in their vaccine, even with an unreliable inactivation process, much fewer people would have contracted polio from the vaccine. It is important to highlight that even though the 1955 vaccination campaign caused some cases of polio, it still managed to reduce the total number of polio cases in the United States about tenfold. This is a huge number of children who didn't die or become paralyzed. Furthermore, the polio cases were quickly identified and the defective vaccine lots from Qatar laboratories were recalled before many more children could be injected with them. Not even 6 months had passed when the production problems had been fixed and a safe vaccine was under production. Nowadays, polio is pretty much gone from the US as well as from most of the rest of the world. Still, the damage was done. Unscrupulous journalists spread the story far and wide, making many parents afraid to vaccinate their children against polio. As a result, the polio elimination campaign took much longer to succeed, and the number of children who died from polio in the following years after the incident is several times larger than the worst possible estimations about how many polio cases Qatar laboratories were directly responsible for. These children could have been saved by the vaccine, but they were not, due to their parents' fears of vaccinating them. Even worse, starting with the Qatar incident, greedy lawyers have started suing pharmaceutical companies for damage that they could not possibly have foreseen, or even caused for that matter. It is debatable whether Qatar should have known better than to blindly trust governmental guidelines and protocols and instead taken a deeper interest in assessing their process using their own methods. What is not debatable, though, is the fact that these court cases have opened the gates for thousands of frivolous lawsuits against pharmaceutical companies for damage that they couldn't possibly have caused. For instance, there have been so many lawsuits against Dow Corning for supposed diseases caused by breast implants that by 1992 they have costed the company over $4.2 billion with a B. And even though not a single study at the time, or until now for that matter, has ever demonstrated that there is any risk associated with their breast implants, the company had to file for bankruptcy. After the thalidomide incident, which will probably get an episode of its own, Many unscrupulous lawyers started suing the manufacturer of the morning sickness pill Bendectin, accusing their product of causing birth defects. Even though there have been absolutely no studies linking the morning sickness drug to any increase in the risk of birth defects, the jurors were finding the company guilty so often that it was a better choice for the company to simply stop producing the medicine. And as a result, every year many women still suffer and a few even die from morning sickness without any treatment being available. Pharmaceutical companies are nowadays completely unwilling to invest research money on products for preventing miscarriages and morning sickness in pregnant women, because they would certainly be sued for any birth defect that happened, even if there was absolutely no shred of evidence that their medicines caused them. Indeed, about 2% of all pregnancies naturally result in birth defects for no identifiable reasons. And a number which is so large that even if only a very small fraction of those mothers decided to sue pharmaceutical companies, it would be for sure more than enough to bankrupt those companies. And as a result, every single pharmaceutical product nowadays comes with a warning label not to be used by pregnant women, regardless of the complete absence of evidence of any risk of harm. This leaves soon-to-be mothers living in the pre-scientific era nowadays. And a few people have also successfully profited from the scientific ignorance of the legal system by claiming that every possible thing, including seat belts and heavy lifting, caused their cancers. Still, I would argue that the worst result from the Qatar incident was, by far, the loss of confidence in vaccines and in the pharmaceutical industry as a whole, which has costed the lives of vastly more people than polio ever did. We are now witnessing a worrisome increase in easily preventable diseases such as measles and whooping cough that could easily be prevented using cheap, safe, and effective vaccines that were actually invented almost a century ago. Vaccines are cheap and very effective because they require only a few doses to result in lifelong protection against horrible diseases. And this is a problem for society because it also means that the pharmaceutical companies that manufacture vaccines make very little money selling them. And because of the legal liability risks, vaccines could cost a lot more money than they make for the companies that manufacture them, due to frivolous lawsuits that are not supported by any shred of scientific evidence. Of course, you should not worry about the financial health of the pharmaceutical industry. As a matter of fact, these companies they have never been so profitable as they are nowadays. But instead of making vaccines, they are investing on cholesterol-lowering drugs, treatments for diabetes, impotency, hair loss, and all sorts of chronic diseases. Not because they are less likely to be unfairly sued for those types of products, but these treatments are very expensive for the patients and health insurances, as they may have to last for several years, which generates enough revenue to cover any potential frivolous lawsuits. No, we are the main victims of these frivolous lawsuits. We are left with expensive and inefficient healthcare options nowadays. Vaccine-preventable diseases are on the rise in many countries, not only because of misinformation and the general scientific illiteracy of the population, but also because large pharmaceutical companies are no longer manufacturing vaccines as they did in the past. For instance, in the United States, there have been severe shortages of vaccines against diphtheria, tetanus, pertussis, measles, mumps, rubella, pneumococcus, influenza, and chickenpox. And different countries have dealt with the situation in different ways. My native country, Brazil, for instance, produces several types of vaccines in state-run laboratories, which are provided for the population free of charge. And this is particularly important here because our population is much more vulnerable to disease outbreaks than the people in richer countries because of our tropical weather and the general lack of proper sanitation in many regions of the country. In developed nations, on the other hand, the government began limiting the legal liability of vaccine-producing companies because of their critical role in public health. India and China now provide vaccines to a large part of the developing world. Personally, I do not know how to change this situation. On one hand, companies should indeed be responsible for providing safe and effective products for the population. On the other hand, there has to be a limit on how easily people can successfully sue pharmaceutical companies so that they keep producing affordable vaccines for the population. Perhaps governments should step in and produce imported vaccines themselves. I don't know. The solution will probably be different from country to country. What is clear is that the Qatar incident had far-reaching consequences on public perception about vaccines, public health, and governmental oversight. As a result of this incident, pharmaceutical regulation has evolved significantly, and it is now extremely rigid in order to assure that pharmaceutical products are safe and effective. It is also important to remember that people involved in this tragedy were people like you and I. Jonas Salk felt directly responsible for not pressing the companies hard enough to make a mathematical model of their inactivation process. Politicians blamed each other, the National Foundation for rushing the production, and the Laboratory for Biologics Control, which gave authorization for Qatar laboratories to sell their vaccines. Indeed, every single person from the Laboratory for Biologics Control that was involved in the polio vaccine program was fired, many of whom couldn't possibly have known the results of their actions. Qatar Laboratories paid millions for the ensuing lawsuits, and the Qatar Laboratories employees were exhaustively investigated by the FBI for potential treason, even though almost every single one of the employees from Qatar had injected themselves and every one of their close relatives with their vaccine before the vaccine was ever sold to the public. These people, they really believed in what they were doing. I hope to leave you with a sense that science is a human endeavor. Mistakes are made, but people have the best intentions and work really, really hard to make sure that things go well. Especially now, during these trying times, we need science more than ever. It is easy to vilify those faceless companies and governmental agencies and to imagine that they are completely devoid of any trace of humanity. But they are made by people like you and I, just try to make the best they can with all of the available knowledge. I am recording this episode from my home, hoping that the scientists will create a vaccine against COVID-19 so that I can finally go out. Science and the pharmaceutical regulations have evolved a lot since the 1950s, and the scientists are much better prepared nowadays to create an effective vaccine in a very short time. It remains to be seen whether these efforts will be successful. You, who are listening to this recording from my future, now know more about the COVID-19 vaccine than I do now, from my present, which is your past. One thing is clear though, the only way out of this mess is through, and the only way through is science. This was Francisco from the Disaster Trio. Stay safe, wash your hands, and take care of your loved ones. Ciao, ciao.
This episode was brought to you by the Disaster Trio. Natalia Florek, Francisco Vitor Santos da Silva, and Marta Brocka. If you liked this episode, please share your opinion and suggestions in the comments. Subscribe to our podcast and learn every month about another industrial disaster. Spread the word between your friends, especially those Portuguese and Polish speaking, because our podcast is also available in these two languages. Bye and stay safe.